So I want to thank the organizers, particularly David and Mimi, for inviting me to give this talk, the keynote. And I think this is probably the most people I've ever spoken to at one time. So give me a little bit of a break. And I've had to follow some really excellent talks. EMPS has meant a lot to me, so it means a tremendous amount to be able to give a talk at the 50th anniversary meeting. So when David asked me to give this talk, he's described it as saying covering the last 50 years, the present, and in the next 50 years of epigenetics research. But I'm going to take a step from Dr. Kunkel, who spoke last night, and go a little bit further back than that. So I'll go decade by decade, starting 3 billion years ago. So in the past, really, epigenetics was invented about 3 to 4 billion years ago by bacteria. As DNA methylation evolved as a host defense against viral invasion of DNA, where the restriction enzymes cut the unmethylated DNA that came in from the viral invasion. And that was a really good system with methylation. Now, in the past 500 million years, this system has been exacted by animals and plants. I'm going to talk about animals here, and to serve other functions. Mainly what we're interested in is gene regulation, also suppression of transposons, which is particularly important, and a few other mechanisms, but mainly excitation for gene regulation. And you can see the proliferation of DNMT enzymes and MBD domains throughout all Animalia. But within So within vertebrates, there's been a real explosion. So that highlights the importance of DNA methylation within vertebrates. So what did the ancestral genome prior to the split of vertebrates look like? It was sparsely methylated in CPG sites, and the proto-CPG islands had not yet become defined by their lack of methylation. But after the split from vertebrates, the vertebrates became heavily methylated. Vertebrate genomes became heavily methylated. And then with the heavy methylation of the CPG sites, that led to deamination, as many people in this room are aware of. That is the most rapidly mutating dinucleotide in the genome. So over time, you get a progressive loss of CPG sites. So you get sparse DNA methylation throughout the genome in the modern vertebrate, followed by continuation of CPG islands. Because they're hypomethylated, they don't lose their methylation. And then you have maintenance of methylated CPG sites in the exons, which increases processivity, and they're also maintained because of high conservation. So interestingly, you need to regenerate these CPG sites, which seems to have been done by transposons copying themselves around the genome. So moving to humans, I think we do want to get to humans. What do humans think of this? So Aristotle rebutted the idea of preformation, where you had a homunculus within the sperm of a tiny little man, who presumably had even tinier men yet still in him, to say that he rebutted this idea to say that organisms develop through gradual progression of undifferentiated material. That was later termed epigenesis. A few thousand years later, microscopes really put an end to the idea of preformation. And then August Weismann did an experiment that could have been done at any time in history to show that there is a separation between that germ layer and that somatic layer. And as Carol mentioned, that this organization was founded on changes that occur in the germline. Cut off 5 tails generation of rats and showed that they were all born with tails. Now, T.H. Morgan of fly fame came along and walked that back a little bit by saying that there are 2 factors that determine heredity. So it's not just factors within the germ cell, it's also modification during development. And I found this image online, and I thought it was... So getting to Lamarck, we cannot talk about epigenetics without Lamarck. Jean-Baptiste Pierre-Antoine de Monet-Chalier de Lamarck was his full name. And I don't think there's too many people with names like that anymore, but you knew he was destined for greatness. He coined the word biology in the modern sense. Let's not knock him too much. But he also put forth the idea of inheritance of acquired characteristics, which was not his original idea, but he really pushed it. When you search for him, you'll see the canonical example of giraffes and a dead giraffe. Now, moving on to modern times, 150 years ago, Metrick discovered DNA, didn't think very much of it. McCarty and Avery proved it's a heritable molecule. DNA methylation was discovered 2 years later by Roland Hotchkiss, who discovered 5-methylcytosine in calf thymus using paper chromatography. This is the actual figure from his paper, which is really cool. So that little bump right there is the first, this epicytosine is what he called it, 5-methylcytosine. That is the foundation of all of us who study DNA methylation. That's the original graphic. In 1981, Compare and Plummeter proved its role in gene expression and regulation definitively for the first time, which wasn't that long ago, maybe a little bit before my time. So there are histones, going back again, 3.5 billion years. They were discovered by eukaryotes, invented or evolved. And what I like this slide because it shows that the histone H3-H4 complex is very similar to the archaeal histone, which only contains a smaller histone, but you can see that it lacks the tails, where most of the modifications are. So that shows the deconservation of the substrate for the histone modifications that many of us in this room are interested in. So getting back to humans, where did we come along? 1884, histones discovered. 1960, Alfred and Mirsky suggested modifications could control gene expression, but it wasn't really. So in 69, there was conservation between plants and animals shown by Delange. I don't know why people keep using calf thymus. Maybe someone in this room can tell me this. But it wasn't until really the 90s, many people thought it was still inert packing material. So that's pretty recent. And these are some great graphs. These are actual micrographs of actual chromatin. I really like to get a sense of what I'm looking at. I think seeing the picture of the phages, for example, is really telling. So both variants and modifications, variants of histones and modifications, can carry epigenetic information. So Conrad Waddington, I think everyone has seen this image many, many times. That is a visual representation of his idea of the epigenetic constitution and epigenotype, in which he really had the first modern definition of epigenetics, which is the product of genotype and the environment coming together to bring the phenotype into being. But Robin Holliday, who I think most people and everyone in this room should know, really had added an important aspect to this definition to make heritability a necessary part of epigenetics. So he said the study of changes in gene functions that are mitotically or meiotically heritable that do not entail change in DNA sequence. So that was a key change to Waddington's definition. So following the progression of definitions over time really gives it follows the science and leads us in the fields to what we're really learning about epigenetics. And we can see the early definitions study mitotically, meiotically heritable changes that are not explained by DNA sequence. Was modified by Adrian Bird a little bit later in maybe a little bit more less specific manner, but structural adaptation of chromosomal regions to signal or perpetuate altered activity states. These two definitions really came together in 2009 by Shelley Berger et al. In Cold Spring Harbor to present a working definition, which is what many people in the field use now. This is the canonical current definition of an epigenetic trait, which gets back to Adrian Bird's altered activity state, is a heritable phenotype and it must be heritable, because otherwise it's just phenotypic plasticity to environmental conditions that results in changes in a chromosome, without alterations in DNA sequence. So the definition is critically important because people tend to just throw epigenetics along as it's just any modification. So we have to keep this in mind. Okay, so talking about some of the past methods. So chromatography was shown, I showed that. Antibody affinity was really important. Peak heights and Sanger sequencing, not a particularly good method these days. Blotting, methyl-specific PCR, and high-resolution MEL were all fundamental in understanding epigenetics previously. Some of these things are really important are still being used and some are not. So Sanger sequencing peak height has been supplanted by pyrosequencing, for example. So the current technologies, this is where we get into the present. So CHIP has revolutionized the study of histone modifications. ChIP-seq, so it's CHIP combined with next generation sequencing, has opened the door to genome-wide region-specific modifications, which many of us do in here. And bisulfite conversion has done the same thing for DNA methylation. So I've always used bisulfite kits. I'm told by some of my predecessors that this was a much more laborious procedure. But kits for CHIP and kits for bisulfite sequencing make life easy now. RNA-seq is doing the same thing for small RNA. There's an entire zoo of small RNA that wasn't even discovered until recently. So present technologies, RRBS, the costs have plummeted. RRBS can be done for $500. WGBS can be done for $3,000. Some people are saying $2,000 or even less nowadays. CHIP and RNA-seq, it depends. You can do it for under 200 to 400 per sample. We're still using DNase-1 hypersensitivity sites. 3C and high C are methods of chromatin conformation that are really important and that they're starting to be used more often. And amplicon sequencing, come talk to me about it. I have some ideas. Then the small RNA sequencing has really come to the fore. There's many people in this room that are studying non-coding RNAs, mRNAs, piRNAs, long non-coding RNAs that code chromosomes in response to the environment. So what has this resulted in? Well, there are many applications of epigenetics. This is going beyond the basic science. What can we do with it? Epigenetics is really being used primarily in fields of nutrition, toxicology, epidemiology, cancer. The DOHAD hypothesis has been really important in developmental origins of health and disease. So it's early life exposures that predispose you, increase your risk to later life development of disease. And then transgenerational epigenetic effects are really important now, intergenerational and transgenerational. And there are books out for all of these, and I think actually several of the authors of some of these are in the audience. So there are many, many resources that come to the fore. Here's just a list. Some of the really important ones would be the epigenomics roadmap that NIEHS was really foundational in supporting, as well as the WashU Genome Browser. There are news sites, and then there's a million different sites. There's almost as many sites as for small RNAs as there are small RNAs. So what has this led to? In 2018, there were 5,381 papers, including DNA methylation, published. That's one paper published every 2 hours. So just this morning, you're already behind. So get on your literature. There were over 1,000 papers published on histone modifications just last year. And this is a really restrictive definition of histone modifications I'm looking at. There are many journals, specialized journals, that will publish epigenetics, but I should not leave out EMM, which is an important journal that also publishes epigenetic research. So where are we going from here? There are yet more marks. Dr. Kunkel asked a question about other incorporated marks. In RNA, there's M6 modification. There's new marks, serotonylation. There's a new histone modification. There's new technologies such as improved sensitivity. When I was doing some methylation work on mice, the old methods of using mass spec required 10 milligrams of DNA. If you want to do tissue-specific methylation in a mouse, you have to grind up the entire mouse to get that much. So you can't do it. Now with improved sensitivity, it opens doors to looking at tissue-specific or even cell-specific changes. 5-hydroxy-methylation, which I've not mentioned, is also becoming more important with TAB-seq. And then direct detection of methylation, hydroxy-methylation, and other marks is coming to the fore, which will eliminate the need for bisulfite conversion and open the door to looking at other DNA adducts directly on the DNA molecule through nanopore sequencing. It's really exciting. Epigenetic drift, germline exposures, transgenerational epigenetics, and environmental epigenetics are the fields of the future. I'm going to go at single-cell genetics. I'm going to go through a few of these just quickly. Here's an example of what serotonylation looks like. and M6. Important definitions are going to be, definitions are going to be important in the future when we look at epigenetic drift. Epigenetic drift was originally thought to be just the change, the wholesale change in methylation as we age. Now we're understanding that there's a natural change and there's a deflection of drift. And that deflection can be more important at different stages of life than others. So this is a really foundational paper by Frog et al. Showing the deviations in methylation between twins in early life is very low, and in later life it's much greater. And then we can see this in aging as well. That epigenetic clock, which was really invented by Steve Horvath at UCLA and has been expanded to many other species since then, can be altered by environmental conditions, which I think is what we're really interested in here. The future germline exposures, there's a special issue in EMM, which talks about this. So there's a Jill Escher's paper, I think is a call to action. It's really important on discussing germline exposures. We're all aware of that here. And transgenerational epigenetic inheritance is becoming important for both germline reasons, aging, and a number of other purposes. And the Target 2 consortium in IHS is critical in explaining some of these exposures that impact the epigenome. So I think the last two slides here is the DNA methylation clocks and aging is going to be a really important issue. As Carol mentioned, maternal aging, we need to look at maternal aging, we need to look at somatic aging, different tissues. We see differences in different tissues' aging rates along with environmental exposures. It's important for our society. So the future, conference, textbooks, non-Mendelian is becoming mainstream. The NIH is explicit about including epigenetics. The interest is holding steady if you look at a Google search here. And I just want to point out in the boxes here, you might wonder why every year there's a massive dip in the search for epigenetics that occurs between May and September when students are out of school. So when they come back, searches go up. So I think that means that the teaching is going well. So anyway, thank you.